Hi, I'm Bill Bialek. I'm a theoretical physicist. I teach at Princeton University, and I want to talk to you today about some of the challenges that we face at the interface between physics and biology. I think many of you know that there's a lot of excitement at this interface because more and more of biology today is becoming accessible to quantitative experiment. So there's a classical descriptive biological tradition, but more and more of the phenomena that we see in the living world are now susceptible to the kinds of quantitative, large-scale experiments that are the norm in the physical sciences. And this transition has created a lot of excitement. It's produced an enormous amount of data. And in areas of biology that range from the behavior of single molecules all the way up to the dynamics of large populations of organisms, we've seen sort of independent revolutions in every subfield, where the whole character of how we approach the phenomena of life is changing. And what I want to talk to you about today is this transition from a classical descriptive science to a quantitative science to being able to summarize what we observe in mathematical models. And finally, I want to talk about the challenge of reaching one step further, to doing the kind of thing that we're used to in theoretical physics, but has not really happened yet in biology. And that's to have more than just models for individual phenomena, often with lots of parameters that you have to fit in order to get all the details of the data right, but rather having theories that are a bit more grand in scope and have the power to predict what you will see when you go out and look at the world from some more basic principles. And it's this search for a kind of unifying mathematical language that I think has been characteristic of physics, and which, if you said that you were interested in doing this in a biological context a generation ago, well, for example, when I was a beginning graduate student, people looked at you with a lot of skepticism, to put it politely, that it just didn't seem that biology was susceptible to that kind of theorizing. And this revolution on the experimental side, I think, has changed everything and has made the search for that kind of theory in the sense that we understand it in theoretical physics, a real research program rather than just a fantasy about how we might someday be able to understand things. And so the way I'm going to try and introduce you to this evolution of our understanding and then the challenge that lies in front of us is to take a very specific example, which is the control of gene expression, in particular the transcriptional control of gene expression, and which is something that's happening in every cell in the living world all the time. And I'm going to dig in a little bit to that example and show you where I think the challenge lies. And then at the end, I'll zoom out and note that many of the things that we're saying about that particular example really are things that you could be saying about a much wider range of biological problems. So that you'll see that this challenge of building theory is something much more general. So the example is transcriptional regulation. You remember that every cell in your body has the same DNA. What makes the cells different is that they read out different genes to make proteins. This is happening not just in us, but in single-celled organisms as well. When bacteria find themselves in different environments, they read out the information coded in different genes to make different proteins that are useful in those different environments. And that process is controlled at many levels, but one of the control steps is right at the point where you read out the information that's coded in the DNA to synthesize messenger RNA. So what you should keep in mind is a stretch of the genome, a piece of DNA, and along this there's the gene that is coding the coding region for the particular protein that you're interested in. You know what's going to happen when this information is read out during transcription. There's a large transcriptional apparatus that sits down on the genome and starts to walk along the DNA. And as it does so, it spits out a molecule of messenger RNA that finds its way out, and eventually, of course, is transported to the ribosome where it's translated into protein. You can regulate the amount of protein that you make at many stages. One of them is just to regulate this process. So there are also binding sites along the genome that sit here close to the point where transcription is initiated, where other proteins, which are called transcription factors, which I'll just call TF, can bind. Of course, there are many of these proteins also floating around in solution. And when these proteins bind, you can imagine that if they bind and get in the way of the transcriptional apparatus, then they will repress the expression of the gene. Alternatively, they might bind in such a way as to make it easier for the transcriptional complex to find its way to this start site and get going in the process of transcription. So you can have transcriptional repressors, you can have transcriptional activators. Now, this simple geometrical picture is not bad if we're talking about bacteria. That's where it came from. By the time you get to complicated organisms like us, this is probably an oversimplified cartoon, but it's nonetheless the thing that people keep in mind. And the result of this process is that if you think about how much of the gene you're actually expressing, and you plot that as a function of the concentration of the transcription factor proteins in solution, then if this is a repressor, so it gets in the way of transcription, you expect that as you have more and more of the transcription factor, you fill up more of these sites and block transcription more effectively. And what you'll see is that transcription falls as a function of the transcription factor concentration. Alternatively, if this is a transcriptional activator, then you expect that as more and more of the transcription factor is present in solution, more of it binds, and it makes it easier and easier for the transcription process to get started. And so you see something that looks like this. Now, in the old days, this is where you had to stop. But now you can do much better. You can reach into the cell, you can engineer the genome so that transcription factors have attached to them a fluorescent protein. And so you can look at the cell under a microscope and measure the amount of fluorescence, and that will tell you what the concentration of the transcription factor is. Similarly, you can engineer the gene that's being read out also to have a fluorescent protein attached to it, perhaps in a different color. And then you can measure not only the concentration of the transcription factor, but also the expression level just by measuring the fluorescence. Now, convincing yourself that that's a reliable measurement and really proportional to how many molecules you have, that takes work, but that's been done in many cases. And so these relationships, these sort of input-output relationships between the control signal and the output of the transcriptional process, or the output of the whole process of gene expression, has now actually been measured quantitatively in many examples. And one of the things you learn when you do that is that even if you find two cells that have the same concentration of the transcription factor, or even the same cell at a different moment in time comes back to have the same concentration of the transcription factor, what you see is that the actual level of gene expression fluctuates. And that fluctuation, if you do enough work in the experiment, you can convince yourself is not coming from noise in your measurement. It really is variability in the behavior of the cell itself. And so these pictures should be associated with little error bars along the curve. And those error bars really represent noise in the process of gene expression and its control. And we understand there are very basic physical reasons why this happens. Individual molecules behave randomly. They take random walks through solution and find their way to their binding sites. There aren't very many of these molecules altogether. If you look inside a bacterial cell or the nucleus of a eukaryotic cell, you'll find that the total number of transcription factor molecules might be of order 100. And it's not only, the cell can't decide to count all of the molecules inside it. Those molecules have an impact only when they find their way to this binding site right here at this very specific point along the genome, which of course is maybe altogether 100 base pairs out of millions in a bacterium and orders of magnitude more in eukaryotes than in us. And so this very small number of molecules behaving randomly, the randomness doesn't average out the way you were taught in your high school chemistry course. In high school chemistry, we talk about the concentrations of molecules as if that was a pure number. But really, there's a limited number of molecules in any small volume. When you do experiments in a beaker that has a liter of material, you don't notice this. But when you look inside a bacterial cell, which is 10 to the minus 12th, a thousandth of a billionth of a liter, then these effects become really, really important. And that's where these error bars come from. So we've been able to push to the point where we have mathematical models that describe this process. Essentially, you take this picture seriously. Molecules bind and unbind from their binding sites. When they bind, they influence the binding of the transcriptional complex. The transcriptional complex moves at some rate. It synthesizes messenger RNA molecules. They're degraded at some rate, and so on. And when you're done, you can actually produce models that will describe this behavior. And really, there's three parameters, although there's lots of things that go into determining this. There's three things. There's the overall level of expression, which has to do with how fast you can make the messenger RNA molecules, how long they live, how efficiently they're translated into protein. There's the scale on the x-axis here. How much transcription factor do you need in order to turn the process on halfway? Ultimately, that's controlled by the binding, the strength of binding of the transcription factor molecules to these particular sites along the DNA. And then there's the steepness of this curve. And that really reflects how many of these molecules cooperate. If one transcription factor molecule binding drives the whole process, then this curve will be very shallow. But if in order to turn on transcription, you need 3 molecules to bind at once, or 5 or 7, which is what you see in real examples, then this process can become very steep. This curve can become very steep, and the system can go almost an on-off switch. Now, we went from description to measurement to mathematical models that can describe this curve. We can even understand where these error bars come from. So what more do we want? Well, the problem is that these models have parameters in them. The halfway point along this curve is determined by how tightly the transcription factor binds to this site. But how tightly the transcription factor binds to this site depends on the structure of the transcription factor, but also on the sequence of the DNA in this particular region. The same transcription factor could also be controlling a different gene, and you would discover that this halfway point is in a different place because the sequence along here is different. So that means that these parameters are not fixed forever. In fact, they evolve. And they can evolve rapidly because they're in the regulatory regions of the genes. And in fact, it's even worse than that. The transcription factor proteins themselves can be modified. They can be phosphorylated, they can be modified in other ways, which also affects the strength of their binding. So these parameters are things that have been chosen by evolution. They've been chosen by the physiology of the cell. And so if you have a model that ends by having these parameters, you've left open a big question. Why are these parameters what they are? What is it about this particular choice that's advantageous for the organism? And that's a problem not only in describing one of these processes, but normally these processes are embedded in a network. There's many genes. A single transcription factor could affect many genes, but those genes themselves could code for transcription factors, and they could influence one another, or even feed back to influence the first transcription factor. And if you took this model seriously, then every one of these arrows is associated with that whole set of parameters. And so what we need to know is, what is it about the setting of these parameters that allows this whole network to do its job? And so people are exploring many different ideas that would be candidates for a real theory that would explain why these parameters are chosen the way they are by evolution, by physiological adaptation. So one idea is that what you want to do is to be sure that the expression levels of these genes carry as much information as possible about what the cell cares about, whether that is in the control of metabolism, and you're worried about the concentration of various nutrients in the environment, and you need to capture information about that in order to be sure that you're doing the right thing, or you're in a developing embryo, and you want to capture information about where you are in the embryo in relation to your neighbors so that you can adopt the correct fate in relationship to the rest of the body plan. What's great is that this question about how information flows through a network is something that you can think about not just at the molecular level, inside a single cell. You can think about it in a whole population of organisms. You can think about a flock of birds all agreeing to fly in one direction. What interactions do they have to have with each other, and how do those need to be tuned in order to be sure that on the one hand they all agree to fly in the same direction efficiently, but on the other hand they can be reliably responding to stimuli from the environment like the approach of a predator. And at an intermediate level, you can think about this in networks of neurons, where you have these same kinds of input-output relations for the individual neurons. What lives on the arrows are not these molecular processes of the control of gene expression, but rather the synapses or connections between neurons. And the strengths of those connections, the parameters, if you will, along these curves, are themselves changing during the course of learning. And so you can ask, what is it that the network is learning to do by adjusting these parameters? What's the bigger theory that tells you how all of this fits together? So we don't have these theories yet. We have candidates for them that lots of people are trying out. And the whole process is being driven by the fact that our experimentalist friends can now make these incredibly precise measurements. And the result is that we are at a moment where the hope for theory, in the sense that we use that term in physics, the sense of a powerful and often quite abstract mathematical description that can reach down and predict the details of experiments with great quantitative precision, that hope for a theory is now something that we can turn into a real research program rather than being the fantasy that it was in previous generations. So I'll stop there. Thanks.